Welcome to a Bioinformatics Let's Play. The class I am teaching this semester is a junior level bioinformatics class. This is a computational biology class which students are learning about sort of classic algorithms in bioinformatics. But in addition, they're also learning how to program a bit in order to become sort of competent in data analysis. However, because of coronavirus pandemic, my university is currently closed for on-ground teaching. Instead, all of our teaching has moved online. And because of that, I am teaching over Zoom. However, Zoom is not a good format for the type of class that I'm teaching. So I'm going to be pre-recording a video so they can see me program and see the sort of the lesson that we're going to go through. And so I figured I would upload this to my channel as sort of a bioinformatics Let's Play because it is on coronavirus. So the topic we will be covering today is a bit of coronavirus bioinformatics, how to download data and how to process it. So we're going to be looking at some coronavirus sequences and eventually computing a phylogeny and seeing any sort of inference we can make from this. So this is just going to be sort of sequences that have been generated and are available on NCBI from the SARS coronavirus 2 data set. What you're looking at now is an R console. R is a statistical programming language that is popular in science for doing data analysis and statistics. And so today I'll be showing you how to use R to download and analyze data from coronavirus. The first thing we need to do is install some prerequisites. So the students in my class won't need to do this because they've already installed the prerequisites. But if you are watching this and you're not in my class and want to follow along, I will show you how to install our prerequisites, assuming that you have a sort of a fresh R installation. And if you want to know how to install R, I will link to my video on that below. So first thing we're going to do is we're going to install tidyverse. Just going to type install packages, tidyverse. We're going to need to select our 0, or we need to select 1 just to choose the cloud mirrors. And then this is going to download and install tidyverse. So tidyverse comes with a lot of packages, so when you download it on a fresh install, it's going to take longer than this. And so the next thing we're going to do is install BioC Manager. This is useful for the bioconductor packages. Then we'll do install packages again, but this time we're going to do BioC Manager. So BioC Manager is sort of the installation manager for the bioconductor suite of packages. So this is a set of packages that are used in bioinformatics to load large biological data sets, and we'll be using one of those packages today. So the first thing we're going to do is install BioC Manager. The next thing we're going to do is we're going to use BioC Manager to install bioconductor-based packages. So that's BioC Manager install version equals 3.10. We're going to set up 3.10 for R. And then the next thing we're going to do is we're going to install biostrings. Install biostrings. So we're going to install biostrings, which is a package in BioC in Bioconductor that is useful for loading sequence data sets, which we'll be using today. So I'm going to install biostrings, so this is going to compile the package and set it up for loading into R. The last package we're going to install is IPS. So IPS is a um, program, is a library for phylogenetic analysis. It allows us to call some external tools from within R. This is going to be important later on when we're calling math to align our sequence data. And so after we install IPS, we will be good to start with our analysis. Now that we have installed our prerequisites, the first thing we're going to do is load the libraries we're going to use today. We're going to use library function, library, and we're going to load two libraries. First is the tidyverse package. So this is going to load multiple libraries, and then we're going to load a library for loading sequence data. That's biostrings. So library tidyverse, library biostrings. All right. The next thing we're going to do is we're going to download our data set. Function is download file. URL is going to be https ra.artwrite/SARS coronavirus 2 FASTA-GZ. You got to specify destination file, so the name of a file locally, and this is going to be SARS coronavirus2.fasta.gz. This is a compressed FASTA format that contains nucleotide sequences from the SARS coronavirus. And so let's take a second to look at what this file looks like. Here is the contents of the file we downloaded. This is a FASTA formatted dataset. FASTA is a sequence dataset in which you have the first line lines that begin with a greater than sign are the header line, so it's usually a sequence name here, NC 045512. This is a sequence identifier followed by meta information. And following that is a series of nucleotide sequences, A, T, C's, and G's. And this is the genome of the reference sample from China of SARS coronavirus 2. And so there are lots and lots of lines here. And if we keep going down, paging down, we will see, I can jump ahead, we have multiple samples. So in this file, we have a total of, turn forward, we have a total of 170 sequences. Okay. So now let's go back to R and continue our analysis. Now that we've looked at the file, let's continue our analysis. The first thing we're going to do is we're going to load this file into R. And the way we're going to do that is we're going to create an object called DNA data, and we're going to use the function called read DNA string set. This is from the biostrings package. And now we're going to do the file name. So SARS-CoV-2 fasta.gz. So this is going to load all the information into an object called DNA data. So let's sort of look at what this object looks like. What we can see is DNA data is a DNA string set, which is a group of DNA sequences. It has 170 sequences in it. We have a column here in the output that specifies how long a sequence is, the sequence content at the beginning and the ends of the sequences, and the names that we saw earlier. Now that we have loaded our data, the next thing we're going to do is parse the meta information that is in the header lines. So to do that, we're going to create an object called meta. We're going to pass names DNA data. So this is going to be the the set of sequence names from our data set. We're going to pipe that to a function. So then this is the pipe operator from tidyverse. And so what the pipe operator is going to do is this is going to take the list of names and send it to a function called string split fixed. And string split fix, we're going to fix on a literal vertical bar with n equals 6. So what this line is going to do is it's going to create an object called meta that contains a matrix in which every row will be for one of the names, and it'll have up to 6 columns of the contents of these names split on their vertical bar. So this is going to be our meta information. Now we can look at it by typing head meta. And what we see here, so head's going to return the first part of the file, so the first 6 lines. We see we have a column for the contents, column for the ID, for the location where the sequence was sampled, the host of the virus species, and 2 more columns that we won't use today. So the next thing to recall is if we look at meta column 1, so this is our sequence identifier, we have an extra space in all of our sequences. So we want to fix that. We want to remove that space. And to remove the space, we're going to do meta column 1 is equal to meta column 1 height to string trim. So what this function does is it's going to remove the extra white space in meta in the first column, and we can see that by looking at the column again, and it's now missing. So this will help make things look better later. The second column of our meta object, the meta comma 2, this contains our sequencing locations. So this is where the sample was taken, the country of origin. And so now we're going to tabulate this vector to find out sort of how many do we have from each location. So to do that, we're going to use the table function. So table meta 2 will give us our counts. So what we see here is that we have 2 samples for which we have no location data. 7 from Australia, 2 from Belgium, 1 from Brazil, 60 from China, 1 from Finland, India, Iran, Italy, Japan, working our way down, USA, so there's 54 from USA. I would like to take a break here and hope all of you are doing fine with the coronavirus pandemic, staying inside, practicing social distancing, keeping 6 feet away from other people, washing your hands 20 times a day, and I hope all of you are doing okay and have the things that you need and are able to go out when you need it and are keeping yourself safe and all your loved ones safe as well. The next thing we're going to do is start by renaming our sequences. So we're going to do names, DNA data. We're going to name them after our trimmed metadata. Okay. We're going to do names, DNA data is meta column 1. And if we look at our names and DNA data, see that we now have all of our sequences named after our sequence identifiers. The reason why we're doing this is going to make it easier in downstream analysis to improve our data, to maintain our data. Now that we've renamed our sequences, let's look at the width again. The width of DNA data. This is going to return us a vector that contains the length of our sequences. What you see is that we have both full genome data, so SARS coronavirus is about 3,000 nucleotides long, its genome. And we have sequences roughly that long in our database, in our data set, plus some partial sequences. So these partial sequences are fragments, maybe a particular gene or section of the genome that has been added to the database. But today we're only going to work with our full length data. In order to do that, we're going to create an object called DNA. to hold our full length data, and we're going to create this by doing DNA data width of DNA data is greater than 2900. What this is going to do is this is going to select all the items in DNA data that have a width greater than 29,000. And we can verify that by looking now at our width of DNA and see that it now contains only full length sequences. So our data is made up of nucleotide sequences. So these are A, T, Gs, and Cs that represent the nucleotide composition of our genome. So the sequence of nucleotides encodes proteins, and those proteins are what make the virus work. Same in virus genomes, bacterial genomes, human genomes, animal genomes, plant genomes, microbial genomes, sort of all you have sort of roughly the same encoding, and it's the sequence at which the A's, T's, C's, and G's are in. But one thing that distinguishes genomes is their composition. So what fraction of the sequences are A's, T's, C's, and G's? The next thing we're going to do is we're going to look at the composition of our sequences. So we're gonna create some data set in which we've taken every sequence and generated what fraction of the sequences are A, T, C's, and G's. And so to do that, we're first going to split our nucleotide sequences into vectors. So to do that, we're going to create a object called DNA list. And that's going to be, we're going to take DNA and we're going to pass it on to string split function. And with a pattern of. So what this function is going to do is it's going to take every sequence in DNA and split it into individual nucleotides and return that as a list. So this will be a list of vectors of nucleotides. If we look at the names in our DNA list, we will see that it is actually empty. This is because string split didn't pass on those names when it created the DNA list object. So the next thing we're going to do is we're going to clean up this list by passing the names, by setting the names in the list to be the same as in DNA. Now if we look at names DNA list, we see that it matches, but it contains all of our sequence names. So within DNA list, the DNA list is an actual list, and to access the first object in that list, we want to use DNA list double bracket 1, and this is going to return us our first element of the list, which is a vector of nucleotides. So this contains every single nucleotide as individual characters. T, A, C, G's. Okay. And then if we wanted to count those, we can use our table function from before. So if we do table DNA list bracket 1, we have the nucleotide content of the first sequence in our data set. So that means the first sequence has 8,954 A's, 5,492 C's, 5,863 G's, and 9,594 E's. So this is for our first sequence, and what we want to do is do this for all sequences. And to do that, we're going to use the map function from tidyverse. And so what the map function is going to do, so first we'll do nucleotide. Nuke is DNA list mapped to table. additional DNN option is new. So what this is going to do is it's going to pass the DNA list object to table, but it's going to do every element in DNA list is going to get passed to table. So we're going to be able to get individual tables for each element in the list. And we have this extra DNN equals nuke option is here, and that is to allow some stuff later on to work correctly. And so when we press this, we can now look at nuke, and we see that each sequence in our data set now has a table of nucleotides associated with it. And the next thing we're going to do is we're going to take this table of these individual tables of nucleotides, and we're going to combine them into one giant data frame where each row contains a sequence and each column is for a nucleotide. So we're going to use the map function again to convert our nucleotides, our tables, into a data frame. So we're going to first create a object called nucleotide tab long. This is the long form table. So that's going to be created by typing nucleotide to map DFR. So this map function will create a data frame by combining rows. Now we're going to do as tibble to convert our data, our tables into data frame, and we're going to specify the .id equals name option, and this is going to create a column in our result that passes the name of our sequence along with it. So if we process this and then look at nucleotide tab long, you'll see what I mean. So this is now a tibble in which we have one column for the name of the sequence, one column for the nucleotide, and another column for the counts. And so the next thing we're going to do is convert this to wide format. So this is long format, in which we have two columns for our data and our counts. And what we want to do is we want to convert this where we have a row. Each row, it will be a sequence name, and then we'll have one column each for the nucleotides, and then with count to contain the data. So this is known as a wide format. In order to create nucleotide tab wide, we're going to use the pivot wider function. So we're going to type nucleotide tab long to pivot wider, where with the option names from nuke, so we're using our nuke column to get the names from, and values from n, so our count column. So when we do this, you will see that nucleotide tab wide is in the format we wanted. So this now has one row per sequence with columns for the different nucleotides. However, some of the nucleotide characters, some of the ambiguous nucleotide characters are not found, and those are NA. So what we want to do is run our function again, but this time we want to add a default value instead of having NA, so we're going to make this 0. So to do this, we add an option values fill list n equals 0. And what this is going to do is for any n for which there's a missing value, so in NA it will use 0 instead of NA in our object. So if we look at that and then at our wide values, we now have a nice clean data set that counts our nucleotides along a row and measure, and then has one column per nucleotide. Next thing we're going to do is look at our column names. So if we go names, nuke, tab, wide, this is going to return our column names. We have one column for name, columns for A, C, G, and T. It's our standard nucleotide. We have a column for N, which is an ambiguous nucleotide representing all possible characters, and additional ambiguous nucleotides. So sometimes when we are generating when we're generating genomes, we don't know, I can't exactly figure out if it's an A, C, G, or T, and so that is represented in data sets using alternative characters. And so it's important. So this, you can kind of think about this as sort of missing data or ambiguous data. So the next thing we're going to do is count up the missing data in our data set with the goal of removing sequences that have a whole lot of ambiguous nucleotides. Now we're going to create an object called nucleotide ambiguous. And this is going to be based on having nucleotide tab wide, selecting only the columns for ambiguous nucleotides. If we select minus A, C, G, T, this will return all of the columns in nucleotide tab wide that are not A, C, G, or T. So this minus within select tells it to return everything but the 4 listed columns. If you now look at nucleotide ambiguous, you can see that we still have a name column for the sequence and all of the ambiguous columns. Now what we want to do is we want to sum up these rows to get a total count of how many ambiguous nucleotides are per sequence. And the way we're going to do that is we're going to do nucleotide ambiguous count. It's going to be nucleotide ambiguous. We're going to remove the name column, so we're going to do select again, minus name. And then we're going to turn that into a data matrix. So this will be now, instead of a data frame, this will be a matrix of counts for which we can now do row sums. Okay. So once we have this, we can now look at our nucleotide ambiguous count and see we now have a vector of the count of ambiguous nucleotides. And what we see is that we have a set of sequences that have a lot of ambiguous nucleotides, over 1,000 here, over 10,000 here. And so we're going to use an additional filter to remove sequences from our data set that have a large nucleotide ambiguous count. So the way to do that, we're going to create an object called DNA filtered. And that's going to be based on DNA, where nucleotide ambiguous count is less than 10. So we're going to select everything that has less than 10 ambiguous nucleotides. And if we look at DNA filtered, now we have 100 data sets left. So far, we have downloaded coronavirus sequences, inspected our meta information in our sequences. We've looked at our nucleotide content of our sequences, and we've used all that information to clean our data set. The next thing we're going to do is we're going to generate a multiple sequence alignment of our data. So multiple sequence alignment will tell us how our various sequences are potentially related in the sense of what sections of the genome correspond to one another. And because these are close sequences, we don't expect there to be any sort of complex differences between them because they're all closely related from the same outbreak. And then after we have a multiple sequence alignment, we will be generating a phylogeny that will tell us how our sequences are evolution related and may give us some clues to sort of different strains or different infection points in our data set. Okay, so now we are back in R, And in order to generate our multiple sequence alignment and generate a phylogeny, we're going to first load two libraries. First one we're going to load is library ape. Ape is a phylogenetic toolkit for R, and the next library is IPS. IPS is a library that allows us to run several external tools for phylogenetics. The external tool that we will be using shortly is MAFT. MAFT is a multiple sequence aligner. It is a really good multiple sequence aligner. I use it a lot in my own lab. And luckily, IPS contains a function for running MAFT. Now, MAFT is external to R, so you need to have MAFT installed on your machine for this part to work. And we're going to first start with saying generate an alignment object with MAFT, find the MAFT function. We're going to convert as DNA bin our DNA filtered object. So we're going to convert DNA filtered object to a DNA bin object, which IPS and this MAFT function can do. We're going to set quiet equals false, so we see the mapped output, and now we have to specify the location of our mapped binary. In my machine right now, it's in USR bin mapped. And so when we run this, this is going to call an external mapped program, and it's going to align our sequences. Luckily, our sequences are very similar, so even though there's many of them, this actually happens pretty quickly. We're now going to save our alignment, the right DNA function, and so we're going to save it to SARS CoV2 ALN.FASTA, and with the format FASTA. So this will save our alignment to a local file, which we can then use to visualize in an alignment visualizer. This is a program called CView, and we're visualizing our alignment in it. This is what our alignment looks like, where all the sequences have been aligned such that similar nucleotides are in the same column. And we have, in this format, every nucleotide has a color. So A's are red, T's are blue, G's are yellow, and green. So it quickly allows you to see patterns. And so what the alignment did is it took our real data, which is our nucleotides, and added gaps such that we have similar patterns in our sequences. And as is common, sort of the beginning of sequences and the end tend to have more gaps, and this simply has to do with data availability at the end. Not really, simply not biological. Meaning, the one thing we can do here is change our setting to by reference. So now this allows us to actually compare our sequence to the reference and see how they differ. In this mode, anything that matches the reference will be a dot, and this allows us to quickly highlight where there are potential mutations in our different sequences. So here's like a couple of these. We can scroll down. We have 100 data sets, and we can scroll over, and these mutations in our data are potential spots that allows us to reconstruct the spread of this virus. So a lot of viruses are identical to one another, and so those are usually very closely related, and ones that have a couple mutations in them are more are sort of track the spread of disease. Things that share common mutations compared to the reference tend to be related. And so if you have two individuals that have the same virus that has the same kind of unique mutation, you can potentially make an inference that those individuals were infected similarly from a similar source. There's actually 30,000 nucleotides in here. I'm not going to go all the way through, but this gets an idea of what virus looks like. Now that we have our alignment object, we can generate a distance matrix between all of our sequence. And so we're going to call that result D. D is going to be the distance of DNA using the alignment data with the model of F84. So F84 is a particular evolutionary model, and we're going to use that to construct a distance matrix between all of our sequences. If we do that, we will have our distance matrix, and if we look at it, we see that we have a distance object in which we have our rows. Here's a good one. So now you see that we have, say, the distance between MN908947 and MNM938348 is 0. But between these two, it's above 0, so there's some mutation between these two. And so the idea is that sequences that are closer together in evolutionary time will have closer to 0 distance, and ones that are farther apart in evolutionary time will have a farther distance. And the advantage of having these distance matrices is that we can use it in a method called neighbor joining to generate a phylogenetic tree. So we're going to do tree is nj. So this is neighbor joining from the distance matrix, and we're using neighbor joining because it is fast and there doesn't require any external programs in R. So now we have a tree object that is unrooted, and so this phylogenetic tree with 100 tips and 98 internal nodes, labels. So we're going to root on this, which is the reference sequence, and so we're going to do that by making a rooted tree. Our tree is going to be the rooted tree tree rooted by NC 045512. And so what this is going to do is generate a tree in which our sequences are an outgroup, and we can sort of plot this tree. And it looks like this. Let's see if I can scale it up. So we have a phylogenetic tree in which our sequences are labeled at the tips, and you can see because this is sort of an outbreak, we have a whole bunch of sort of sequences that are very independent of one another. So this is what is known as basically roughly a star cluster, and individual sequences. This doesn't tell us too much because these are sequence IDs, so we're gonna change it and we're gonna plot it so these have some of our metadata. In this occasion, we're going to use the location of the sequences. Now, in order to rename the tips of our tree, what we're going to do is we're going to create a meta subobject. So we're going to subset our metadata so that it only contains the rows that are still in our filtered data set. So it's going to be meta sub is going to be meta, and we're going to select all the columns in which all the rows in which the first column, which is our sequence ID, can be found in row name of the alignment. So we're going to select all rows in meta for which the first column of meta is in the row names of the alignment. So this is going to give us all the meta information from our remaining sequences. From this, we can rename our tree, tip labels to meta sub. Once we do that, we can now plot. We plot our tree again. plot our tree again. We now have a new tree for which the sequences are labeled by their country of origin. And some of the interesting is that although we have approximately similar number of sequences from the US and China, clear that sort of China is more broadly distributed, which makes sense because it is the origin of SARS coronavirus originated in China. And we see some sort of interesting sort of USA clusters coming here. And so how you read this is each tip is a sequence, as a label. As you go back, two sequences will combine, and that's an estimated common ancestor of those two sequences. The vertical distance doesn't matter, only the horizontal distance does. And things back here at the base are all match the reference sequence from China. And so this is sort of not really enough data to really understand the outbreak fully, but it is sort of interesting with the public sequences that we have available to see certain patterns. And it's kind of interesting. So I think that is a good place to stop for today. As a recap, we have downloaded SARS coronavirus 2 data. We've curated it. We've extracted metadata. We've filtered it and aligned it. Looked at nucleotide composition, and we've constructed phylogenetic trees. So all sort of important central bioinformatics skills, and I hope that you have learned something from this. If you have any questions, please don't hesitate to ask in the comments below. If I've forgotten anything, check the pinned comment. I'll make sure all that information is there. And I'd just like to thank you for watching, and be safe out there. Be safe with your family. Keep your friends and family safe, and good luck. Good night.